photosynthetic limiting factors. 13.4 limiting factors. Limiting factors. Limiting factors of photosynthesis. Now, this is exactly ordinary level IGCSE knowledge. Limiting factor. There's no other limiting factors being introduced. But at this particular level, you have to be more informative about what each of these limiting factors do, how they are affected, and specifically which stage of the cycle or phosphorylation is affected by these limiting factors. Okay. Now the three main external factors. The three main external factors affecting the rate of photosynthesis are, let me write that, that's photosynthesis, okay, are number one, light. Please remember these in this particular order and it will be much easier to move on from one to the next. Light intensity. Light intensity. Number two, carbon dioxide concentration. Now, I would write it like this, carbon dioxide concentration, if you were IGCSE. Since this is a A2 level thing, this is what I'm going to do. CO2 bracket. Square brackets represent concentration. We know that, right? So if I have sodium chloride within this, we're talking about the sodium chloride concentration. If we have H plus within this, talking about the H plus concentration. And like so and like so. Okay? CO2 concentration. The third one being, this is a more obvious one, temperature. All three of them are obvious ones, yes, but it is temperature. We will start off by discussing about light intensity. Number one, the first limiting factor being light intensity. Light intensity. Light intensity, very shortly speaking, I will use two arrows. If light intensity goes up, rate of photosynthesis goes up. Very simple. We'll talk about why, and I will give you a graphical analysis as well. First things first, we'll write the concluding statement. So the greater the light intensity, the greater the light intensity, the more energy supplied to the plant. Obviously, the more light energy supplied to the plant. So, therefore, simply, the light dependent reactions, faster light dependent reactions occur. Light dependent reactions can occur extremely fast. More light, more photoactivation, more electron excitation, the more faster the light depends. So what are we talking about? What do we mean the light dependent stages? Non-cyclic and cyclic photophosphorylation. You see, this hitting of light and subsequent photoactivation happens a lot. Happens faster, more frequent, with more energy. Therefore, we will have an overall faster light dependent state. Right? So what is the result? So what do we get? Now, if we have a faster light dependent phase, what's the result? Thus, more NADPH and ATP are produced. These are the only things we're taking from the light dependent stage. So more NADPH, more ATP is produced. Much more of them are produced. So in turn, the Kelvin cycle can also occur at a greater rate. That's it. Why can it occur at a greater rate? Because we're producing more NADPH, more ATP. These are the things utilized in the Kelvin cycle. So if we have more of them, we can have a faster Kelvin cycle as well. Now let me draw a very simple plot to denote how light intensity behaves with the rate of photosynthesis. So if we have x, y, simply speaking, we'll talk about the rate like this. So this will be the rate. This will be, so in this case, light intensity. So this is rising. So as we increase light intensity, the rate increases. Makes sense, right? This makes sense. So in a perfect world, if we are providing more and more and more light, this is what should happen, right? In a perfect world, that is exactly what should happen. The rate should completely increase. But we hardly live in a perfect world. This happens. Even though we provide a lot of light, this curve eventually plateaus. We call it that the curve plateaus. Curve plateaus. It falls. Why though? Why? We are giving enough light, so as we go to this direction of the graph, we are giving more and more and more light. Plus, plus, plus light. A lot of light. But even though we get a lot of light, why does the curve plateau? So now this part makes sense. In this area, light intensity is the limiting factor. Why? Because around this area, we don't have that much light. So this is a small area. If I label the graph 5, 6, 7, and 4, here we are just giving 1, 2, 3 units of light. So the rate is growing, but light intensity is also around here. We have 10 light intensity, 10 units, 10 arbitrary units of light intensity around this area. But still, the rate is not increasing, it's plateauing. The reason being, around these areas, we have enough light. That's the thing. Around these areas, we have enough light, enough and more light. So what happens? Some other factor, whatever it is, some other factor becomes limiting. That's what happens. What are the possibilities of some other factors? Now we only have 3 factors. Now let's see. So light intensity is not limiting at that point. What can possibly be limiting? It's either CO2 or temperature. Those are the other factors that might be limiting. So CO2 or temperature. Simple. CO2 or temperature. That's it. These two things can be limiting around this area. So this is the effect of light intensity on the rate of photosynthesis. Overall, greater the light intensity, greater the rate of photosynthesis. Greater the rate of photosynthesis. Moving on to the second one. What color did I use for that? This thing. Nope, I just got rid of that color. Wait here. Carbon dioxide concentration. Let's see how CO2 concentration affects the rate of photosynthesis. How does carbon dioxide concentration affect the overall rate of photosynthesis? So that is exactly what we're measuring with all these limiting factors, okay? How does a certain factor affect the rate of photosynthesis? That is what we're trying to measure. I'll give you the overall hot like I did before. So higher carbon dioxide, higher rate. Simple. Let us see why. Now this is required for more like in, yeah, for or in the Kelvin side, which is like independent stage. Fixed by, you should be able to answer this right now. This would be Rubisco, the enzyme Rubisco. To combine, what does it combine with? What does CO2 combine with? Combines with RUBP. Combines with RUBP. So simply put, thus, if more carbon dioxide is present, faster this step of the Calvin cycle can occur. That simply means that the overall rate is going to simply increase. Overall rate of photosynthesis. Excuse the little bit of cursive mixing. I'm just writing it very fast. Yeah. When I look back at my handwriting, I am having second thoughts, but yeah. So simply more carbon dioxide, more fixation by Rubisco, more of the initial stage of the light independent stage, and thus a faster Kelvin cycle and of increasing the rate of work. So that's the thing with these limiting factors. Whenever you are presented with a limiting factor, you have to know what stage or where exactly this thing affects. And since you know the Kelvin cycle, since you know photophosphorylation, that will not be a problem. It will not be a problem because you can link it back. Now, if I say light intensity, we should be talking about the light dependent stages, right? We should be talking about non-cyclic and cyclic photophosphorylation, right? So faster the light dependent reactions can occur, non-cyclic, cyclic. And then when we're talking about CO2, we absolutely know that we're talking about Rubisco fixation and the Kelvin cycle. More carbon dioxide, more fixation, more of that metabolic pathway being active, therefore a faster Kelvin cycle, so faster rate of photosynthesis. Just like I did with that, I mean, light intensity, I'll give a short graph. It's the same exact graph. So boom, in a perfect world, boom, that's what should happen. Not a perfect world. Right, so around this area CO2 is limiting. Around this area, some other factor is limiting. Now, in this case, what can that some other factor possibly be? Now it has to be one of the two others, light intensity or temperature. Or temperature. It has to be one of those two factors. Now, not carbon dioxide concentration. Now, do you see? Here we're talking about light intensity. So light intensity is the limiting factor. Here we're talking about CO2 concentration, so CO2 is a limiting factor here. Here, some other factor. So two of those other factors are CO2 and temp. So in this case, we're talking about carbon dioxide concentration. So the other factor has to be light intensity and temperature. Hope you get my point. Right? So some other factors, you can't write CO2 here. Because in this case of the graph, we are talking about CO2. We're talking about CO2, right? We are simply talking about CO2. Okay, let's go on to the third one. Third and final limiting factor, temperature. This is an interesting one because now carbon dioxide, light, you can directly link to certain stages. Temperature, you really can't do that. This is an overall thing. Now, whenever we talk about rates of reaction, temperature is always predominantly there, no matter what. Organic reactions, inorganic reactions, biochemical reactions, pathological reactions, inflammatory reaction, temperature is always there, and temperature will always affect all of these stuff because of consequent kinetic energies and the interactions and all of that. Right. So now when it comes to temperature, I'll give the overall one as well. Higher the temperature, higher the rate of photosynthesis. And using that, we can conclude something like this. All three factors. If I increase, rate goes up. If I increase, rate goes up. If I increase, rate goes up. Okay. It's a conclusion. An overall conclusion. Now let's see how temperature does its job. D as photosynthesis. As photosynthesis is an enzyme controlled reaction. We know plenty of enzymes and coenzymes and co-transporters and all these stuff are included. Right. Reaction and increase and increase in temperature would increase the rate, would increase the rate. But once again, up to an extent only. Got it? Up to an extent only. What is this extent and how does it work? So, if the temperature is increased above the optimum, we know what happens to enzymes. They begin to enzyme denaturation occurs. Now these two graphs, they looked exactly same right over here. They look precisely the same. I mean, basically the same. So light intensity increases, that happens. Carbon dioxide concentration increases, that happens. Kilodos and light. Temperature is not like that. That's why I told that temperature is a special, a rather special limiting factor. So if I were to assess that, it would look something like this. Doesn't have to be even that long. Right. Temperatures increased, rate increase. Bro. Now, what you would expect is the curve to plateau like this. But here's the deal. So this is the optimum temperature. This is the optimum temperature. Once we go beyond the optimum, what happens? Enzymes denature. So they completely change their active site and then they can no longer metabolize. So the rate drops to literally zero because the enzymes are destroyed. So the rate drops to zero. Rate temp. Now this graph, I am certain that you have seen this a million times before. This is simply the typical enzyme denaturation. This is the typical enzyme denaturation right over here. We go beyond the optimum, we will lose the active stages of the enzymes as the protein becomes denatured. The protein becomes denatured. So that is basically all of the limiting factors and how they affect different stages and the overall stages of photosynthesis and how they interact with each other. Moving on to the final part of photosynthesis that we have to discuss. This will be the 13.5. The experimental part of photosynthesis. Now, I will start off by explaining about DCPIP and methylene blue. Might sound like gibberish, but it's simple. DCPIP, DCPIP, and methylene blue, methylene or methylene, yeah, methylene blue are redox indicators, that undergo a color change. Color change. These are two different indicators, okay? They undergo a color change. That color change being, or is a more vibrant blue to write the color change. Okay, that. So these are both blue. They are known for changing from blue to colorless. That the blue I used. Oh my god, I don't think you can see it right. Okay. Blue to colors. Blue to colors. Right. Now the thing is, these DCPIP and methylene blue compounds, they are artificial, they are artificial H plus and electron acceptors. Electron acceptors. So if they absorb or accept electrons or H plus ions, they undergo this color change. So simply speaking, we can use them to keep track of any reaction that produces H plus and electrons. For instance, photosynthesis. Photosynthesis, right? So simply speaking, no matter what, we calculate the rate of, it's very similar to this. Now, the rate of photosynthesis, which is what we are calculating in this case, is equal to 1 over time taken. So time taken is measured in seconds, so that therefore the rate becomes s minus 1. Now, how does that happen, you might ask? So it's like this, no, 1 over s, oops, 1 over seconds, 1 over seconds, right here. We need to get the unit for rate over here, so the s should go up. Now it's to the power 1. When it goes up, it becomes s with a negative 1. Okay, that is how that becomes s minus 1, right over here. The rate of photosynthesis is equivalent to 1 over time taken. 1 over time taken. Now, in brackets, I will give you a small explanation that will help with this artificial H plus and this thing accepting thing. Okay. Now, since the light dependent reactions in thylakoids, thylakoid membranes actually produce, what do they produce? Electrons. They are picked up by this DCPIP or methylene blue. methylene blue, and hence they undergo color changes. So simply speaking, what are we doing? We are treating this DCPIP and methylene blue as indicators in this case. We are treating them as indicators in this case. Once we treat them as indicators, we can use them to calculate the rate of photosynthesis, calculate the rate of how much this reaction happens, the rate of this reaction, this particular reaction, which is photosynthesis, the rate of any other reaction which involves H pluses and electrons, E minus. Okay. Now, using this little bit of information, we can develop a very small experiment to find out or to figure out the effect of the three limiting factors on the rate of photosynthesis. So can be observed by the below scenario or the below apparatus, I'll call it. Now I will use green color to do this. So it's very simple. The apparatus is very simple. We have, I don't know, whatever it is, a lamp. Not an artist, by the way. I'm trying my best to draw them. That looks like a, I don't know. Have you watched War of the Worlds? So it looks like one of those machines. Never mind. Lamp. Looks like a stage light. Never mind. And then we have a specific distance. How do we measure that specific distance? We actually use a meter ruler. Meter ruler. So it's the big meter ruler. So obviously the lamp is producing light. And then here we have a beaker filled with water. Then we have a test tube stoppered by a bung, and that bung is connected to a gas syringe. Like so. It's connected to a gas cylinder, like so. And here we have a plant inside. So this plant's rate of photosynthesis, that is what we'll be measuring. I'll use a yellow color to label it. So this is an aquatic plant, otherwise we can't use it in this case. Aquatic plant. So inside we have water. And we have sodium hydrogen carbonate. We have sodium hydrogen carbonate to absorb the carbon dioxide produced. Okay, to absorb the carbon dioxide produced. So the meter ruler, this is a gas syringe. If gas is pressing on this plunger will move back and you can obtain a reading from these measurements, right? Okay, and if you want, you can label this as well. This is just a delivery tube, also known as a capillary tube. Capillary tube or delivery tube. Yeah, right. Capillary tube or delivery tube. And this beaker also contains water, but this time it's not for the plant. So this is a thermostatically controlled. I'll just call it a temperature controlled water bath to keep the temperature at an optimum constant level, right? So this is what happens. The lamp is switched on. Obviously, light shines on the plant, right? Then photosynthesis happens. Now, what exactly is the method that we are utilizing here to calculate the rate? This is the method. The method would be the volume of oxygen gas collected per unit time. If you paid attention to this arrangement of a pattern, you can clearly see we have a method to collect some gas. We have a method to collect some form of gas here. Now, what gas does photosynthesis produce? What gas is photosynthesis very well known for producing? That is oxygen, not carbon dioxide, okay? That's respiration. We produce carbon dioxide in respiration. In photosynthesis, we produce oxygen. So the lamp, now this lamp is actually controllable. You have certain intensity. So you have intensity 1, light intensity 2, light intensity 3, 4, 5, and so forth. You can control the intensity and slowly increase. Right? So when the lamp is switched on, light will be produced like so. It will hit the plant. The plant will realize that, okay, we have light. We can start the light-dependent stages of photosynthesis. So photophosphorylations occur, and you produce ATP and NADPH. Calvin cycles occur, and then we produce all of that. And then finally, all the products of photosynthesis are produced. So starch is stored, and we have oxygen being produced here. And this oxygen being produced is going to go through the capillary tube to the gas syringe, fill the gas syringe, and make the plunger move back. Make the plunger move back. So you will be able to obtain a reading here on the volume of O2 gas collected. On the volume. So, gas syringes, they have, what do you call these? These are calibrations. These are calibrations. So you will have 1 cm3, 2 cm3, 5 cm3, like so. These will be calibrated lights. This will simply be calibrated lights. So, for example, if I were to carry this experiment out, I will keep this apparatus for 5 minutes of time and measure the volume. So, for example, okay, now I'm keeping this for 5 minutes at light intensity 3, I'll say. So I'll be keeping that for 5 minutes at light intensity 3. At light intensity 3, here is marked here, 1, 2, 3. Light intensity 3, right over here. And then I will be obtaining a certain set of results within this 5 minutes. So I'm just going to let it run. So what this happens, oxygen is produced, oxygen is pushed through the plunger and through the capillary tube and onto the gas syringe like so. The plunger is pushed. So after that 5 minutes up, I will look at this gas syringe in the proper way and take a read. So for example, let's just say we have produced 20 cm3 of oxygen. Oxygen was produced in this time period. It's produced in this time period. Therefore, I will now be able to calculate the rate of photosynthesis. I'll be able to calculate the rate of this particular photosynthetic reaction. Let's try that. So, I produced 20 cm3 of oxygen. So the method that we are using is the volume of oxygen. Now go back to volume of oxygen collected per unit time. That is how we calculate the rate. Something 1 over time take per unit time. So time is 5 minutes. Now the thing with time is this is not the SI unit of time. It has to be seconds. So to convert minutes to seconds, what do? You multiply by 60. 5 times 60 seconds. So let's just see what we get here. 20 divided by 5 times 60. Now let's say 5 times 60 should be 300. if I 22, 1, 0, 0.056, no, 6, 6, 6, 6, 6, 6. I think it's a recurring. Let me just double check if I calculate. Oops. Okay, yeah, 0.0666. I'll just call it 0.07. 0.07 cm3 per second. S minus 1, or you can just throw in a slash per second. 0.07 cm3 per second. Now you can calculate it as cm3 per minute. 20 over 5, that would be 4 cubic centimeters per minute. That is also possible, but let's just convert. Now, honestly, if we convert to exact SI units, we have to convert the cm3 to m3 as well. That's the thing. Typically speaking, the question will give you in these cases what units you should use, whether you could keep it in cm3 per minute or cm3 per seconds or some other complete. Now, if the question states convert to proper SI units, you have to convert cm3 to m3 because that's the SI unit, meter cube, and time of course seconds. Okay, the question will guide you on to what units you should technically follow to obtain an required result. Right, now this was for 5 minutes at light intensity 3. Now I'm gonna keep for 5 minutes at light intensity 4. And now I saw that I produced 30 cm. Let's calculate the rate. We'll just do it in cm3 per minute this time. So rate is equal to 30 cm3 minutes, 5 minutes. You see, I have not changed the time. 30 over 5, 6, right? Yeah, I think so. Yeah, 6. 6 cm3 per minute. Now let's just compare those values. 4 cm3 per minute to 6 cm3 per minute. What has happened? Has the rate increased or has the rate decreased? By the looks of it, 6 is greater than 4, right? So I can clearly see that the rate of photosynthesis has increased as I have increased the light intensity from intensity 3 to intensity 4. Now, are these results concordant? Are these results cohesive with what we talked about? Let's see. Let's go to light intensity. Right. Now, if light intensity increases, here, light intensity increases, we concluded that the rate of photosynthesis has to increase as well. Rate of photosynthesis has to increase as well. Has that happened? I think it has, right? I think it has. I bumped up the intensity from intensity 3 to intensity 4, so that is more, that's increased, and as a result, 4 cm3 per minute has increased to 6 cm3 per minute. So an increase. So there's an increase in the rate of photosynthesis. There's an increase in the rate of photosynthesis, and therefore our results are concordant with whatever we predicted in our hypothesis, and this experiment is a true experiment. One last thing I should mention. So now from going from here to here, I only increased the intensity. I kept everything the same. So the temperature of this water bath was the same. I used the same exact plant. I used the same exact duration. Nothing changed, right? I only changed the intensity. Why? Because that is the only variable I should be changing, because that is the only variable I am investigating. So I'll just draw a box here and say, so when investigating one variable, in this case we're investigating light intensity, you have to make sure you keep everything else the same, others constant. Have to ensure to keep the others constant. have to ensure to keep the others constant. Right? In this case, for change in light intensity, we have to keep temperature, time, the plant, all that stuff the same. Okay, all that stuff the same. A noteworthy mention would be that sometimes this intensity is measured by the distance. So if you keep the lamp closer, like around here, the intensity is high. If you keep it behind, the intensity is low. Sometimes that approach will also be used. So do take note of that as well. But it's the exact same procedure. So you have different intensity, you're following the same exact time period, and everything else would be the same. You calculate the rate in the exact same fashion, and you will be obtaining a specific set of results. That is the same scenario all over again. Going back to the first part, not here, right here. So this is where we started. The photosynthesis light independent stage of the Calvin cycle. This is the entire Calvin cycle. This is a very shortened version of the Calvin cycle. These are all the substrate, all the intermediates, and these are all the organic and inorganic molecules involved and produced, and all the enzymes and carbon dioxide levels, and how the products of photophosphorylation reduce light. Everything is discussed under the Calvin cycle. Then we talked about the limiting factors, the three main external limiting factors. That's important: light intensity, carbon dioxide, and temperature. Overall conclusion is all three of them are increased, rate increases. That's an overall thing. Okay, if all three of them increase, the rate would increase as well. Now, the greater the light intensity, more energy, so blah, 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 all of that. Carbon dioxide, Rubisco, Calvin cycle is affected. Temperature, enzymes, okay, that's the only thing you have to remember. In temperature, we have to talk about enzymes. Temperature and enzyme controlled reaction, in this case, photosynthesis. Therefore, it follows the typical enzyme denaturation curve. And the experimental part of photosynthesis, where we calculate the rate. In this approach, we are using volume of oxygen collected per unit time. And with that, that is the end of photosynthesis A2 levels chapter 13.